Hey everyone, welcome back to the GEP Pathways video series. Today we will pick back up on the last section of Part 6, Part 6.5. Recall that in Part 6.4 we used our spliced RNA-seq reads to confirm the coordinates of intron 1. This same annotation strategy can be used to determine the coordinates for intron 2 between CDS2 and CDS3. So in our Genome Browser, to examine the regions surrounding intron 2, enter NC_052530 colon 19151055 dash 1915156 into the search terms text box and click on the Go button. Zoom out 3 times to examine the 306 base pairs surrounding this position. There are 2 splice junctions predicted in this region that connect CDS2 with CDS3, junction 00113128 and junction 00113129. The T+N alignment for CDS2 ends at 19151055. The potential splice donor site at 19151057 to 19151058 for CDS2 is supported by the splice junction predictions junction 00113128 and junction 00113129, the NCBI ref-seq genes and spawn alignment of D-melanogaster proteins, gene predictors Gemoma, NSCAN, and Augustus, and the RNA-seq data. There is one nucleotide, A, at 19151056 between the last complete codon, AAC, and the potential splice donor site. Hence, this splice donor site is in phase 1 relative to frame 1. The T+N alignment for CDS3 spans from 19151156 to 19151359 in frame +2. The potential splice acceptor site at 19151152 to 19151153 for CDS3 is supported by the splice junction prediction 00113128, the NCBI ref-seq genes and spawn alignment of D-melanogaster proteins, gene predictors Gemoma, NSCAN, and Augustus, and the RNA-seq data. There are 2 nucleotides, CC, at 19151154 to 19151155 between the first complete codon, TTC, and the potential splice acceptor site. Hence, this splice acceptor site is in phase 2 relative to frame 2. This phase 2 splice acceptor site is compatible with the phase 1 splice donor site for CDS2. Click on the junction 00113128 to determine the number of spliced RNA-seq reads that support this splice junction prediction. Here we can see that the score is 5,875. Click on the back button of the web browser to return to the Genome Browser image. In addition to the splice junction 00113128, which supports the proposed splice acceptor site for CDS3 at 19151152 to 19151153, there is another splice junction which suggests a different splice acceptor site, which is junction 00113129. Thus, we need to investigate whether predicted junction 00113129 is supported by other lines of evidence. CDS3 in D-Yakuba includes 2-methionine in frame +2 at 19151255 to 19151257 and 19151348 to 19151350. Hence, the splice junction ending in 129 could indicate the presence of a novel isoform of Reb in D-Yakuba. However, when we assess the number of spliced RNA-seq reads that support the splice junction ending in 129, we find that this junction is weakly supported by only 8 spliced RNA-seq reads. Thus, there is little evidence to postulate a novel isoform of Reb in D-Yakuba based on this splice junction prediction. Note that in addition to the scores, When analyzing multiple splice junction predictions for an intron, be sure to confirm the predictions are on the same strand as the gene you're annotating. For example, if a splice junction is predicted in the negative strand and the Reb gene is on the positive strand relative to our scaffold, we could eliminate that prediction. Taking into account the splice site phases we found in part 6.3 and using parts 6.4 and 6.5 as a guide, you will be able to find the start and end coordinates of the remaining coding sequences for the gene model of Reb PA in D. Yakuba. Remember that we do not include our stop codon coordinates in our CDS5 ending coordinates, and that we must write them down separately.